if you just have 10 animals, you start out with 10 babies and you grow them up. Okay, you're not putting in spawning or recruits. All right, you don't have any inputs at all. You just have the same ones. All right, and that's an issue in a lot of the models, older models in the literature, that they weren't looked at in terms of conserving numbers of animals. Okay. All right, so if you then go and worry about some of these other things that I just mentioned, which is shrinkage due to, say, poor environmental conditions. All right, the blue box here is what I just showed you. All right, then you put in resorption. All right, so now you've done that something like the reproductive tissue to cover the somatic tissue needs during times of bad conditions. All right, reproductive tissue in a lot of these benthic invertebrates is 30 to 50% of the body weight. So when it spawns, the animal throws away 50% of its weight. All right, humans, when we have babies, we get rid of about 10%, you know. So you can imagine throwing away 50% of your body weight every time you spawn, right?
I'm sure some would like that.
Yeah, there are people who might have just, I don't know, I had a baby, I wouldn't want to do that. Anyway, reproductive tissue counts for that. So once you put reproduction in here, all right, then what you end up dealing with is all of this here. All right, you have to keep track of the gametes and where they go, how viable they are, and that kind of thing. And then you produce larvae. And when you produce larvae, you come down here, and then you worry about larval mortality, recruitment events, and all of that. And then the larvae come back and provide new recruits. All right, so that's how you get new numbers of animals. So once you start doing this, you end up with a lot more complexity in what goes on here, and a lot more things to parameterize and to keep track of. And then you add additional terms to the equations here, where now if you can shrink, then the big guys come back, and the little guys gain from that. So you're now moving animals up and down in this size class gradient that we were talking about here. All right, so the kinds of simulations you get from this kind of model are shown here. This is a 4-year simulation. Size classes over here. What's plotted is the log of the number of individuals here over time. And the gray stuff here is the development of the reproductive tissue. All right, so if you look here, there's a buildup of reproductive tissue here. This decrease in the population, remember this is in terms of energy here, or the size. This decrease is the spawn. So you can actually see the loss of the energy, the loss of the mass as the animal spawns. So you can track the spawning events through this whole thing. You also get the range of size classes that are reproductively active, the ones that are producing recruits that go back into the population. So this gives you the time history of a lot of things, gives you the change in the population structure, and that's what you get from this kind of model, and that may be adequate for a number of applications. But the limitations here are that you're looking at the average individual. So it doesn't allow for variability in things like reproductive rates, respiration rates, those kinds of things. The larvae in this kind of model are a loss of mass for the post-settlement population, and then they're basically a gain of mass to the smallest size class. So what they're doing, you're just sort of adding and subtracting mass. You're not changing the characteristics of the population the way you might if you put in larvae that have a range of genetic variability. All right, and the way the model is developed here is if you gain or lose weight through a spawn, it requires that you gain or lose length because it's based upon a weight-length relationship. Right? And that's a big limitation to these kinds of models because it's not realistic. You can lose weight, but not lose length, okay, as an example.
So where does something like oxygen come in? Do you have any limitations?
Oxygen limitations are built into these models here. Right, because these animals live in environments that tend to have hypoxia or anoxia at times of year. So that's a huge one. Okay, so let's go look now at these kinds of models with the recent past. That's what they call here. All right, so now we go to the individual. We put that in here, use an individual-based model, and then take the individual and through some magic develop a cohort by varying characteristics along a range of variability, and then somehow magically take this cohort and build up a population. All right, so what we want to do here is use an individual-based model to look at ranges of variability, build up a cohort where they have individuals that have phenotypic variation, and that variation is determines the population response to the environment. And in this type of model I'm going to show you here is that if age, which is a measure of how old the animals are, is that the age and where length is sometimes used as a proxy for age in these animals, that is decoupled from the weight, right? So age and length are now actually decoupled, right? All right, and so why is that? If you look at how animals grow, and this could be anything, it could be humans, it could be any animal. This is the length axis here, this is the age over here, so this is the age-length distribution. And if you look, the reason that the other one I was showing you has a problem is that if you look at a given age here, you see that that age has associated a range of lengths, right? You look around the room, you find people who are your age, you're not all the same size. So there is variability in length with age. And if you look at length can, go over a span of ages. The same length goes over a span of ages. All right, and that range represents the genetic variability within the population. And you get these nonlinear curves to these age-length distributions because most animals hit a maximum length, okay? And, we all grow that way. We seem to be able to continue to increase in mass, but, length is, or in height in terms of humans is finite. So this type of relationship tells us that, what we were doing in the other models where weight and length are directly coupled isn't a very good way to do things. All right, so now if you look at a model that was developed for a hard plan, what's shown here, again, you go through the thing, you have the environmental forcing here, filtration, assimilation, respiration, get down here to net production, apportion it between somatic and reproductive tissue, and then if the somatic tissue is positive, you can in fact gain weight. If it's negative, you in fact lose weight. But what we do here at this point is use a concept which is scope for growth, which is if you've got enough left over after you've done all your maintenance, you've done everything, then you can increase in length. Right? So this is a condition index thing. So you look at the animal's condition. If it's positive, then it can gain in length. If it's negative, it can't. Or if it's neutral, it just stays at that length. So that's a different approach here. So there's a different calculation that allows the length and the weight to be decoupled. Reproduction still goes through spawns, eggs, recruits, and then back into the cohort. All right, so by doing this, you can actually allow the animal to gain weight but not reproduce.
Is your mortality constant?
In this example I'm showing you, there is no mortality. It doesn't have to be. You can do whatever you want it to do. Right? But there is a mortality here to the recruits. There's a planktonic mortality. There's a mortality that's due to the fact that there's genetic abnormalities in the gametes, which is why you track the gametes. I'll come back to that in a moment. Okay? All right, so in this kind of a model, you still calculate, but you're calculating differently. You look at the time change in the weight, you then go through the condition index, and then look at the time change in the length. So it's a little bit different approach to how you do this kind of model. Okay, so what do the solutions look like? Well, this is an example of a simulation in weight and length space that was done for a hard clam, mercenary to mercenary, if people are interested. Right, the dotted line here shows the absolute maximum length and weight that one of these animals can get. The dashed line here is sort of what's the standard, and this line is the simulation. So it's not a nice smooth curve now as it was in the previous ones. So what it does is you see it going up and down, changing, going up and down, and that type of thing. So you can get an increase or decrease in weight without a corresponding change in length. And if you look at these kinds of things, these are the spawning events. Right, so this animal spawned right in here, that's a big spawning event. There was no corresponding change in length. Right, and that's a big advance, I think, in how these kinds of models can be done. And it's not just these kinds of animals, it's pretty much any organism that you're growing up in these size class models.
What's the figure for spawning? Seasonal?
The spawning queue for shellfish, there are two parts to it. One, there's a temperature, apparently a temperature queue, so that when it warms up. The other is when the animal reaches the point in an oyster when the reproductive tissue is at about 40% of the total body mass, right? And you need those two things to happen, and when they happen, the animal spawns. Okay, right? So if you look at then how do you do something with this kind of model, or how would you use it in the context we're talking about here? All right, well, we can look at the effects of environmental conditions on clam weight and length. All right, and this is a simulation up here of the dashed line here is clam length, this is weight, and this is a sensitivity simulation that was run with a period of starvation imposed in years 3 to 5. Right, starvation would be something like environmental conditions were such that the phytoplankton bloom was delayed, there was lower phytoplankton production in the year, something like that went on in here. So you impose that through the environmental conditions, and then you look at the effect of that on the growth. Right, this is the relative condition of the clam, right, and for the two cases, there's a dashed line and the solid line here, where it's a standard case and then the one with starvation. One thing is that the condition of the animals change over time, and what happens is the animal gets older, it spawns less frequently. There's some biological or ecological implications of that. But the management part of this is that if you look here, all right, this is an animal that's 1 gram, all right, and in this simulation, it reaches that weight and length here in about two years. Turns out this is about the size that they start harvesting these animals. Right, during the starvation period, an animal didn't get to 1 gram until about 1.5 years later. Right, so this model has been used to look at things like what happens under changing temperature conditions, changing food quality, changing food availability, food quantity, and the application of this model was to hard clam populations in Great South Bay, where apparently the phytoplankton community has shifted from large cells to small cells, and the clams grow poorly on the small cells. So it's looking at things like this. So when do you open the clamming season? When do you close it? How do you set the quotas? All right, now, as I said, this is an individual-based model, so how do you take this and go to something that's more cohort and population-oriented? Well, in this case, it's a very ad hoc way of doing it. You take something like a Gaussian function to look at, and you take a couple of characteristics of the individual, like respiration rate or assimilation rate, and you vary them over a range around the results you get from the individual-based model. All right, so you do that, you get this large, you run it's all post-processing in effect, looking at if you have this individual and you know these two biological rates vary in this way, can you sort of fill in a matrix that would give you a range of variability around that one individual? All right, so you do this and you pick the characteristics you want, and you build up a series of cohorts based upon the spawning events, which are usually 1 or 2 per year or a season. All right, so if you do that, you get sort of this kind of picture here. This is just an example, all right, where the respiration rate, the first simulation would be here at 1, all right, where that would be the one individual-based simulation. And then you vary respiration rate, this would be double the value at 1, this is half the value at 1, and then this is the initial